Well, good evening, everyone, and welcome to the Brian Crombie Radio Hour on Saga 960. I've had a bunch of really interesting shows on COVID-19. We've talked to some people trying to do clinical trials. We've talked to people doing DNA tests. We've talked to people producing masks and PPE. And tonight we've got a team from the University of Waterloo that are developing a novel vaccine for COVID-19. And so I wanted to find out what's going on. So, gentlemen, thank you and welcome to the Brian Crombie Radio Hour. Who are you all? Can you introduce yourselves to our listeners, please?
Sure, absolutely. My name is Roderick Slouchev. I am an associate professor at the University of Waterloo in the School of Pharmacy, as well as the CSO of Metaphage Pharmaceuticals as well.
Excellent.
I'm Marco Coyne. I'm a professor of chemical engineering at the University of Waterloo. And our lab really involves the manufacturing of vaccine and vaccine products. Yeah.
Hi, my name is Emmanuel Ho. I'm also a professor at the University of Waterloo School of Pharmacy. My research team is very interested in developing nanomedicines as well as other types of drug formulations to improve drug therapies and for the treatment and prevention of diseases.
Fantastic. Well, welcome, gentlemen. I really appreciate you coming on our show. So tell me, where are you in the status of developing a vaccine?
Well, it's only been actually a few weeks since we got started in this process, and it's been quite fast and furious since that point. So we're at the point where the construction of the vaccine's in process, and the entire process has been laid out on how this is going to occur. It's a very expedited at present, and we expect to even be completed our preclinical trials by as early as the end of 2020.
And the preclinical trials would be how many people?
Oh, sorry, these are preclinical. So these would be the animal tests and animal models that would be done by the end of this year, which we would be working with the Vaccine and Infectious Disease Organization in Saskatoon to do some of the animal testing on the model.
And then tell us the status, what's the process from then? Then it would go into healthy volunteers and then into sick people, or what's the...
Well, there are a number of sort of regulatory processes that have been expedited due to the urgency of COVID-19, and this is still in the process of changing, ever changing, to figure out where it's going to be at any one point. And there's a regulatory feasibility along the lines to be able to figure out how that's exactly going to work. In general, that would, exactly to your point, you'd start with a phase 1 trial whereby you'd be looking at the safety and dosing of what is the potential for the vaccine, after which looking at what are the immune responses that are generated and some of the efficacy around that vaccine in phase 2 and forward.
But then you have to actually find out whether it's efficacious by putting it into people that are then going to be subjected to the virus. I saw a fascinating interview with I think the chair of Harvard Medical School, or a former chair of Harvard Medical School, that said that you couldn't actually give people the virus because it would be unethical to do so, because we don't have any drugs to cure, and so therefore, what do you have to do? You give it to a whole bunch of people and wait till they just happen to get it by chance within the community?
Well, surprising, actually, to that first point, this is a hot topic for discussion that's ongoing right now that is actually considering the concept of challenging humans for the perspective of trials. And the reason for that is just because the amount of data that can be collected is so quick. Of course, on the counter side, the question is, how ethical would it be to do something like this? And that is, of course, questionable. This is something that I don't think you'll see come to light, even with respect to COVID-19. But the idea for vaccination is that we want to understand that it's safe. So what we learned from even phase 1 trials at that point is the sort of dosing around it and what is considered to be safe and the potential side effects that would be dawned on from a virus like this. After that, the idea would be to sort of implicate, to look at the virus and what type of an efficacy profile you generate. And most of this is done with immune responses that can be looked at through the serum of individuals that have received the vaccine, as well as a number of other tests that can be done in that respect.
So sorry, you're looking at the serum of people that have received the vaccine, and then what? Subjecting the serum to the virus?
No. Sorry, the serum would give you what the antibody profile is. So if we're mounting a proper antibody profile that we would expect from a vaccine like this, and the serum can be analyzed, and you would see what are the types, what are known as isotypes, what are the types of antibodies that are being generated, are they neutralizing? And a lot of that can be done out of patient rather than within that patient, of course, for safety reasons.
So let's just be simple. When do you think we'll have a vaccine that can be given to 30 million Canadians?
That's a great question. Of course, that's the question that everyone's asking. It's unfortunately not that simple. The first thing you have to look at is the fact that vaccines are not easy to make. They can take anywhere from 20 to 40, and in some cases 40 plus years, and still not have a vaccine. SARS-CoV-2 is a tricky virus. Similar viruses like this that you'll see for dengue, for Zika, even for HIV. And in the case of how a vaccine can work, we have to be it's not only about the efficacy of the vaccine, but it's also very important that the vaccine itself is safe. And in some cases, particularly for viruses that are a little tricky like this, there's a potential for a vaccine to actually potentially cause harm by the type of immune responses that it generates.
So your answer is 40 years?
So the answer is in potentially as little as, I would say, a year, but potentially no vaccine for several years.
Well, we're having an interesting chat with a team from the University of Waterloo. Sounds like three at least smart people. I was going to put myself in that category for a second, and I won't, but three smart people. We're going to take a break and come back in just a minute with this team from University of Waterloo. Stay with us. Well, welcome back to the Brian Crombie Radio Hour on Saga 960 AM. We're chatting tonight with a group of gentlemen that are all assistant professors or professors at the University of Waterloo, some in engineering, some in pharmacology, and we're talking about a vaccine for COVID-19. And Roderick was just telling us that it could be a year, it could be a long time coming. So Roderick, you know, there's this company in the United States, I think it's called Moderna, that has got a different way of far more quickly creating a vaccine. Are you familiar with that?
Yes, I am.
And tell me, what do you think the chance of that is, and what do you think of their different process?
I think that it's a very interesting vaccine, one that's using genetic material rather than some of your more conventional approaches. I think that there are a number of considerations that were taken into the design of this vaccine, which are appropriate to the type of responses that are necessary for SARS-CoV-2. That being said, it all comes down to what the testing will indicate. So far, so good. I know that the results have proven positive as they're approaching, I believe they're in stage 2 clinical trials right now, or initiating those. But so far, things are looking good. You always have to look at any vaccine from three perspectives. One, of course, what is the safety component? Secondly, is it efficacious and will it work? And third is also the accessibility component. And the accessibility means, is it scalable and is there availability for as many doses as are necessary globally? In all cases, I think Moderna is able to hit that mark. I do believe that it's a promising vaccine. But as I mentioned before, this is a challenging virus. And as much as we can have our fingers crossed that these vaccines will all make it forward, it is important to also be testing a variety of different designs.
There's also a vaccine being developed by Oxford University that's getting a lot of press. Are you familiar with it?
I do believe so. I mean, there are a few out of England as well, so I'm not sure exactly which one you're referring to.
And I think that both in Canada and the United States, I'm not sure if elsewhere, people are talking about actually manufacturing some vaccines on spec. What do you think the government should be doing in regards to supporting development of vaccine?
I think this is a global challenge. So having those types of approaches is important. And I think that these types of global collaborations are certainly things that should be invested in. And this is something, it's an interesting time in that you'll see a number of these collaborations forming. And it's not only a feather in the cap of those who are able to be part of the right answer and the effective vaccine that goes forward. But again, I mean, if we have a number of different scenarios whereby we're able to expedite the process and be able to safely and efficaciously put something forward, then everyone's involved in this, not only nationwide, but globally. So having these types of collaborations is very important. And I'm glad that you're seeing these things happening.
So are you suggesting that these different companies, different universities that are working on different vaccines are working together and they're not competing?
In some cases, of course, they're competing. But on the other hand, I mean, we're competing against SARS-CoV-2. I think what's really important here is that a number of different modalities and a number of different approaches by which we're designing vaccines are being tested. So this isn't only about the SARS-CoV-2 concept. There's a durability component. So even if it may not be able to address SARS-CoV-2, it may become important for SARS-CoV-3 or other viruses and other infectious diseases as we move forward. This is a very rich time on the science side, and new paradigms that you see may be stepping forward for different types of vaccines, and that's just as important.
So tell me, how did you guys get together? How did this project start, and what did the three of you differently bring to the table?
I can speak to how this was initiated. When SARS-CoV-2 became an urgent kind of global pandemic, of course, certain precautions and measures had to be taken. Our university, most of the labs shut down. So as a result, research stopped, or graduate students stopped, and the labs were pretty much quiet. And at some point, the three of us realized that we all work on therapeutic platforms. We have the ability to be doing something about this. And as we discussed the potential for a vaccine, realizing that we each bring different expertise and different therapeutic platforms to the table, from a cross-disciplinary approach, realize that we can actually do something here and design a vaccine that I believe can address some of the, not only the tricky issues with SARS-CoV-2, but seriously make a difference and reach those 3 points of being safe, efficacious, and scalable.
So Roderick, tell me what it is that you've done in the past and what you bring that the others don't, and then maybe the others can speak to the same thing.
Sure, absolutely. My background is actually in genetics and microbiology. I work with bacteriophages, which is one of the elements being put forward in this vaccine design as well. Bacteriophages are bacterial viruses that have a number of applications on the therapeutic arena, very easy to work with, very genetically malleable. And as a result, we can orient them in a variety of different ways. In a vaccine approach, bacteriophages are entities that are present within our body at higher number than our own human cells, and we coexist with them daily. So they offer a number of potential therapies or applications, and in this case, they are a good fit for a potential vaccine because they are very penetrative, can get to the site of infection that we required them to enter cells appropriately, and to even deliver the genetic material, which for us is the payload that is necessary to encode the viral components that generate the vaccine qualities.
Awesome. One of the other two?
Yeah, sure. Our lab really focuses on making lots of virus. Mostly viruses that will become vaccines. And the idea is that we're working not only on the process side from an engineering perspective, but also from an engineering perspective of the biology. And so we're trying to pick out components that exist within these biological entities and bring them together in a way to enhance the production of all these different components. Roderick and I have had a long history. We started at the University of Waterloo essentially at the same time, and we've known what each other has been doing for many years. And every once in a while, our lines of interest sort of overlap, and more so in the last few years. And so this background of us knowing what we're doing and these overlapping areas sort of brought us together, and we're building off of those past experiences and past collaborations to really bring this approach to SARS-CoV-2.
And Emmanuel?
So my contribution to the team is mainly focused on developing a formulation, a potential product that we can translate all of the different technologies that we just heard from Roderick and Mark into a potential product that humans can actually administer. Right? You can imagine, once you have some sort of therapeutic, you have to be able to make it so it's easily administered to humans. So my role then is to potentially develop this vaccine into what we call a nanomedicine, which is basically to package it into what's called like a small little drug carrier system that can hopefully enhance therapeutic efficacy as well as efficiency. Furthermore, as you're aware, we're also trying to formulate this into, for example, an intranasal spray. As you know, a lot of people are really against getting injections, right? So we're trying to develop a more patient-friendly method of administering a product. Furthermore, as an intranasal spray, as you know from those that are infected with COVID-19, one of the ways to test for it is really an intranasal swab, right? So the nose is one of the sites in which the virus first targets before getting to the lungs. So by administering the vaccine at the target site of infection, we're hoping this will also help improve the therapeutic efficacy. So I'm more of the formulation or pharmaceutical scientist of the team.
Awesome. Well, it sounds like it's quite the team you put together. So let me understand this if I could. So what you're working is something you call a bacteriophage. And I thought that a vaccine typically contains a small amount of the virus, and you're putting that virus into people's bodies and they create antibodies to combat the virus when it comes. Is a bacteriophage different than that?
It is. A bacteriophage is an actual bacterial virus. To your point earlier, that is one of the conventional approaches to vaccine, where you would take you would take a virus, you would heat kill it or treat it chemically such that it is essentially non-viable or inviolable, and then use that to be able to essentially stimulate the type of immune responses and not worry about the propagation of the virus. That is a conventional approach that you'll see quite often and has been used for decades for a number of different viruses. In the case of SARS-CoV-2, it's important that the type of immune response that's generated is the appropriate one and most pertinent. And generally, for that type of a response, especially for a viral response or something that is propagating inside your cells, you want to stimulate the right branch of cell-mediated immunity. And so to do that, you would really want your viral components produced intracellularly. The approach that we're taking in that respect is using a bacteriophage, which is just the delivery vehicle. It's really important to state that bacteriophages by themselves are very safe in humans. They do not grow in human cells. They would just sit there and do essentially nothing if they were not properly engineered to carry some type of a genetic cargo or genetic load. In this respect, we are doing something what's known as a DNA vaccine. A DNA vaccine means that it's the DNA or the genetic material that has to get to the target cells and has to be expressed before there is any type of an actual vaccination that would occur. We are using the bacteriophage as the vehicle to be able to deliver what we know to be some of the safest DNA cargo. And they're specialized types of vectors, which by presenting them are not only very efficiently expressed, but are also very safe and protect against any of the potential problems that you would get with DNA delivery. And by delivering this to these cells, we would then have these viral components expressed within our target cells, which would be the cells of the lower respiratory tract, which are the target cells also of SARS-CoV-2. And by doing so, would express something that's known as a virus-like particle. The virus-like particle mimics SARS-CoV-2, looks like a shell of it, but carries none of its genetic material. And the importance of that is that it would be processed the same way that SARS-CoV-2 to stimulate all the right types of immune responses, but would not be able to propagate by itself. So it's a multi-component system which simulates how SARS-CoV-2 would go about an actual infection, but doing so in a very safe process that eliminates some of the immune backfirings, as well as some of the inefficiencies that would come with other more conventional vaccine designs, such as a typical inactivated virus.
So the bacteriophage is entered into the body through this intranasal, what do you call it, a gel or a lotion, Emmanuel?
Sorry.
Go ahead.
Totally within your wheelhouse here.
Yeah, so the idea is it'll be very similar to what people are already familiar with. For example, the seasonal flu intranasal vaccine spray, right? So it would actually be
a spray.
A spray, correct?
Okay, and that carries this bacterial phage, and the bacterial phage carries some DNA to the upper respiratory sites, and in that DNA is the shell of the virus?
That's correct. So the DNA itself actually encodes these viral components that would self-assemble, and the self-assembly of this shell would not only be processed and allow for those presentation of those viral components to mount the right type of an immune response, but would also leave those cells and be able to bind to adjacent cells, thereby potentially protecting them against SARS-CoV-2 infection or binding to those same sites.
So in the end, you're still looking for the body to create the immune response. I presume that's the antibodies that we talked about earlier, the way that the conventional vaccine would do so, but it's through this different vehicle, the bacteriophage carrying the DNA entering into the cells, and the DNA containing this viral shell that you were talking about.
That's correct. And that has a lot to do with the sort of accessibility component to it. We can mass produce how these bacteriophages are made, and they're genetically engineered to package this very safe DNA vector, and at the same time can be targeted directly to these cells. So there's a targeting component to it. There's a very strong
How does it target to the upper respiratory or the lower respiratory system rather than the whole body?
We would be using specialized peptides that are known to have high affinity for the actual receptor protein that would be present on lower respiratory tract cells where SARS-CoV-2 would be binding and where its normal uptake would be taken.
Now, I presume what you're doing now is mostly theoretical. Have you actually got a, what do you call it, a prototype or a product right now?
Not with the bacteriophage so far, so that's still under design and under manufacture at the moment. We do have a version of that with a lipid nanoparticle that's being produced, in which case the DNA vector for the actual viral components is already manufactured, and we're looking at the self-assembly of the virus-like particle at present.
And so let's go back through the process now. So then the next stage would be to do it in animals. When would you propose to be in animals?
The idea is to be in animals within a month to month and a half.
Oh, really? And do we know that COVID-19 impacts animals the same as it does humans?
That's a great question. It's important that the type of animal used is representative of what we could expect within humans as well. There are models that have been generated currently in ferrets, in guinea pigs as well, and in some cases even in mice that carry the right type of receptor that would allow for that targeting or for whatever therapeutic to be properly tested and to look at the type of responses that would be generated.
And you said it would take the rest of the year. Why so long?
Well, there are a number of tests that would have to be done. In our case, it's not just the in vivo test, but also a number of in vitro tests that would have to be done cellularly. We're actually doing all of these at the same time to try to expedite the process. The animal tests themselves will range probably in the area of 2.5 to 3 months. And because we're also testing two elements or two aspects of this vaccine. The first is as a prophylactic vaccine, which means is it preventative against subsequent infection by SARS-CoV-2? But the second element is how therapeutic it can actually be following infection. The idea behind it is that if we're able to generate these virus-like particles, those particles that leave cells should be able to bind to the receptors on adjacent cells, thereby shielding those cells from potential infection by SARS-CoV-2. So we're looking at those two elements that need to both be ascertained.
So what you're saying is that this could be not only a vaccine, but a therapeutic, that after someone gets infected, before it goes rampant through their body, give them this product and it won't go rampant through the rest of the respiratory system?
That's right. That being said, there is a timeline by which that becomes an availability or a possibility for individuals, so that we can mount the right immune response, but also have a therapeutic component that might be built in.
So it almost sounds to me like that would be the easier product to test, because giving such a product to people that are already infected, gosh, the President of the United States was telling people to take whatever it was. So you can imagine that people would be willing to try almost anything versus taking the vaccine and then subjecting yourself to the virus.
Well, as I mentioned, I mean, there are two goals behind this. The first is that there's a therapeutic component, but also the memory and the prophylactic component. So we do want to mount the right type of an immune response because that's what's protective. The second part to that is the durability, and that's a protective against what's known as genetic drift. So as the mutations occur within the virus, and there may be changes to some of the components, that we are able to still maintain protection that would be against, say, SARS-CoV-3 potentially, if there were another outbreak to occur, or even other potential CoVs or other coronaviruses, which may not be SARS-CoV-2, but others that are either related or perhaps even less related.
So one of the things that Tony Fauci in the United States has been warning is that even if we come up with a vaccine, it may not be efficacious against mutations. You think your product will be efficacious against mutations?
This is in the design what we've been trying to do. So the cell-mediated response that we're trying to stimulate first and foremost is the most important against a viral infection of this sort. If we are able to mount response against the most conserved regions, which we're targeting, and which is a component of this vaccine, then we do expect that we will have that durability built in across other variants of the virus following mutations.
Well, this is absolutely fascinating. We're going to take another break and come back with this team in just a minute. Stay with us. Well, welcome back to the Brian Crombie Radio Hour on Saga 960. We're chatting with three gentlemen from the University of Waterloo that have put together a team that are working on a vaccine for COVID-19. I wonder if you could take a step back for a second and just tell me a little bit about how lethal this virus is and how does it actually attack the body? Because you must know a lot about it if you're actually developing a vaccine for it. Roderick?
Oh, sure. SARS-CoV-2 is a coronavirus, which is an envelope virus. And as I mentioned before, there's some tricky aspects to this virus in that it can readily cause inflammation within the lungs and eosinophil accumulation, and something that's known as a cytokine storm. And this is, again, as I point to a number of the vaccines and the more conventional approaches that you would find against vaccines, may not be the best option in the case such as viruses such as this. Now, cytokine storms, what that means is that you can actually trigger an immune response within your body that can result in massive inflammation. And that massive inflammation can do two things. Obviously, it can cause tissue damage itself. And the second component to it, and that tissue damage can lead to serious morbidity or even potentially death in itself. And the second part is that it can actually exacerbate infection itself. And that what that means is that you can facilitate the infection of further viruses into cells. And that, of course, leads to further morbidity, caused by the virus itself. So it's really critical that the right types of immune responses are mounted and that they're checked for these types of immune backfiring, if you will, is the best way of looking at it. So viruses like this are a little bit tricky even in their vaccine design, and of course can cause massive lung damage with individuals that get it.
What is a cytokine?
A cytokine is, the best way to think about it is essentially a hormonal messenger. And these are released by immune cells and can either lead to recruitment of other cells into an area or can activate certain cells, such as B cells, for instance, to produce immunoglobulins, can activate cytotoxic lymphocytes to target and kill cells that they recognize, can lead to other inflammatory cells to become present within an area and lead to further inflammation. It essentially can do a variety of different things depending on what is the function of that one lymphokine or cytokine.
And a cytokine storm sounds like, I guess, the way you've described it, that there's way too many of them and they overexpress and you get too much of an inflammatory response and something bad happens?
Exactly. So it's essentially, it's an uncontrolled innate immune response that can lead to just a variety of cytokines leading to further inflammation and just an uncontrolled response, which can lead to serious tissue damage and morbidity.
Now, if that's what's happening, any idea why it's more impactful on older people than younger people?
I think what you're finding in older people that tend to be more immunocompromised is just the inability to fight the virus itself. And the virus itself is preying essentially first and foremost as its first site of infection being the lower respiratory tract tissue. And so you'll get obviously the lung damage just via the infection that occurs in that respect. That in itself can lead to inflammation and will lead to inflamed responses within the area just due to the infection and the tissue damage itself. But the lack of immune response, particularly in older individuals, can be a serious concern.
Now, you may not know this because it's sort of a little bit out of your area on more on what do you do with it once you get the virus. But any idea why? I've heard that 80% of the people on incubators don't survive. Any idea why the incubation is not more successful?
Well, even once you're on a ventilator, by that point, the lung damage is usually so severe that it's almost past the point of no return. The lung damage that is inflicted by individuals sort of post-autopsy, looking at what is caused by this virus, it can be massively severe. And as a result, the lung damage can just result in inability to breathe. I mean, it's unbelievable, but in a lot of cases, these individuals truly just suffocate.
Suffocate. That's incredible. Mark, maybe I could turn to you. Now, what you were describing is that you're working on something that's called a virus-like particle, and you were saying that this is a protein structure that mimics the coronavirus. Can you explain that?
Yeah, so virus-like particles, I mean, I describe it to my students in the simplest way. It sort of looks like a virus and smells like a virus, but it doesn't have the same abilities as a virus because it can't actually do anything. It just is. And by that, I mean, it doesn't contain any of its genetic material. The genetic material is essentially just an instruction manual, right? And without the instruction manual, a cell can't actually do anything to actually make more of it. And so it's, you buy a piece of furniture from IKEA and you don't have the instructions, it's hard to put it together. Well, probably not really from IKEA furniture, but still, the point is, if you don't have the instruction set, it's very hard to put it together, and viruses without their genetic material can't put together. So what we do is we provide the cell sort of an alternate instruction manual, one that allows it to make certain parts of itself, but not any way of packaging the instruction manual to allow the virus to bring it to other cells. And so one of the things I was telling you about is that from a vaccination point of view or a prophylactic point of view, what we tend to do is make things that resemble the virus, and then we administer that to a person, and they'll mount a defense against that component that we've made. But from a manufacturing point of view, we do this in cells that are typically not human cells. Sometimes they are, but they're not native to the person that is receiving the dose. So what's unique about what we're planning on doing here with the DNA vaccine, or what's unique to a DNA vaccine, is that we're getting, by administering them to humans or to people, we're getting the people to act as their own manufacturing facility. So they will actually be making all those components within their own body. And really, that's the best quality material that you can get. It's the material that's going to be resembling what a virus will actually do within your body, because it's what you are actually producing. You see, a lot of the proteins and components of a virus, they get additional characteristics from the host where they're actually made. And so if you do it outside the body, in a lab or in a manufacturing facility, there's subtle differences to those components that are not the same as what our body would make. And so one of the advantages of our system and the approach that we're doing is we're actually using humans as the manufacturing platform for making the components of the vaccine.
This absolutely sounds very innovative and fascinating. Have you actually accomplished this already with some other vaccine?
So we know that as long as we can get the message inside of the cells, the cell will actually, well, if you get it inside the cell and you can get it to traffic to the right place, those instructions can be read by the cell and those components can be made within the cell. And so from that point of view, we already know we can do that. But whether or not we have the whole trafficking, well, that's when we put the whole thing together with, you know, the formulation and the phage aspects.
Now, tell me about the business aspects of this. So you guys are developing this this vaccine. If you're successful, is it going to be the University of Waterloo that is going to be producing this? Are you going to create a company that's going to produce it? You're going to license it to some big pharmaceutical company? What's sort of the business side of things?
Well, at this point, this is a University of Waterloo endeavor. We are working with two of the spin-offs that come out of University of Waterloo, one called Theraphage that functions, that works primarily with bacteriophages, even as gene delivery modalities, and the other being Metaphage, which works with those specific DNA types of vectors that I was mentioning before. But the most important part is that all of these have combined at present just to be able to expedite this process forward, to make use of all the best partnerships that we can. So it's not just the Metaphage and Theraphage and even the University of Waterloo. We are also working with a variety of other commercial partners to ensure that we have the right type of inhalation device and pulmonary administration device, right type of help with targeting of peptides, and to make that affinity as strong as possible for the peptide that we end up finally choosing. But all of these are sort of coming together to expedite that process as much as possible. And that includes the manufacturing piece as well, although, and it's an important business component to this. And when we talk about accessibility, the ability to manufacture virus-like particles can in itself be quite expensive to make those proteins yourself and to purify those assemblies, versus the actual purification of the DNA, which might be far more cost effective and plausible, and then allow humans to be able to manufacture those VLPs internally within themselves, as Mark was suggesting, essentially serve as their own factories.
Moderna skyrocketed with some of the news that they were developing the vaccine that they are. Are either the two companies that you mentioned public, or are they both private? And are you looking for outside capital, or what's the status?
Well, at this point, they're both private. At this point, there's a lot of public funding just due to the urgency that's arisen due to COVID-19. So I imagine that this is the same, across the world, that you see a number of countries that are increasing public funding to look for new solutions. And those aren't necessarily just vaccines, but also new types of therapeutics that can more acutely deal with the infections of individuals. In our cases, there are a number of public funding sources that we have tapped into and applied for, and the hope is that a lot of the funding can be secured through those routes, and we can again work with a number of the commercial partners that we have in place just to be able to expedite those processes further, in combination with some of these public funds, public groups such as our National Research Council, which could be helpful in the production here as well, and to make use of this. So I mean, this is a far more collaborative approach than you would see for your normal type of business transaction.
And so who do you go to? Health Canada, I presume, for approval of your trials, but is it National Research Council or some other entity that approves the grants that you've been applying for?
Well, a number of the grants that we are looking at are through our engineering council, as well as our health councils for some of the grants that are available. Those are currently being adjudicated at the moment, and hopefully we'll find out on those shortly. A number of other smaller grants which are already been enacted and helping to get things started and to kick off and ensure that we're well underway. But yeah, it's so far. And after that point, from the manufacturing perspective, that would be some of our commercial partners that have already been tapped into to ensure that we have these secured approaches, and of course, moving into the CGMP quality that would be necessary, as well as a scale that would be necessary for some of our clinical trials.
So we've had so much criticism of the government, both Canada and the United States, more in the United States than Canada, about not securing testing equipment, not securing PPE. Is government doing what it needs to do to support scientists like you in developing the vaccine, or do they need to do more?
That's a good question. I think that the best way to preface it is that no one's gone through this before. So this is rather new for governments, just as it is for the investigators going forward. I think what's important is to have that longevity view. This isn't just a fad. This is something that is here to stay for a good while, and it's ensuring not only that we get a solution to this, not only as quickly as possible, but as safely and efficaciously as possible, but also that we are able to push forward some of using this almost as an opportunity to build on the science and to build on new paradigms of vaccines that can help us even further beyond this point into SARS-CoV-3, 4, and other potential infectious diseases that we come across globally. And again, I think the important part here is some of the partnerships that have been formulated, not only even nationally, but worldwide as a result of this. And a lot of the funding that's in place tries to emphasize that. And I think that that's a really important component. This isn't really just even about one country, it's about all countries, and it's a really global effort.
I appreciate you saying that, but frankly, it sounded like it didn't work out that way on PPE. And, we had restrictions on borders. And I've spoken to several different people, both in sort of older industries like autos and equipment, as well as a lot of people that are producing PPE and masks and stuff like that. And they're saying that global supply chains have broken down, and that nationalism was on a rise before this and is now continuing to be on a rise. And it was almost as if the global recession financial response was a global response, but this response so far has been, beggar your neighbor, every country for itself.
I think that's not really within my area. We're not clinicians, so we're not on those front lines to see exactly what's happening. But to your point, I think that there's an acute need which can lead to those types of responses, and perhaps a little bit more selfish in the approaches that could even be seen, nationally or even within the nation. From a science perspective, these are long-term solutions. And I think you have a much more of an opportunity to see some of these collaborations come together. And there's also, as I mentioned, through these collaborations, there's an opportunity for even countries, to put a feather in their cap if they're part of, some of these solutions.
Well, I hope you're right in that regard. I'm still focused, if I could, on how you get it trialed. Whether you just give it to a whole bunch of people and see if the vaccine comes to them, or whether you actually proactively give them the virus to see if the vaccine works. I interviewed a fascinating scientist from Mount Sinai that was recommending a clinical trial of giving people in a healthy setting the active virus, such that they would get the virus, get infected, hopefully recover, and then be immune. So it's almost like getting them vaccinated by getting the virus. He'd applied for ethics approval and Health Canada approval. I'm not sure if he's actually received it as of yet. What do you think the possibility is of actually giving people active virus, whether they have the vaccine or not?
Right, I have come across that as well, and it's a little bit shocking, I guess, when I think about the first virus against smallpox, and the whole concept of where vaccine came from is from the vaccine vaccinia, which was used as a cowpox solution to smallpox, and that's exactly how it was tested. It was given and then challenged with smallpox and found to work. I think we've come a long way since then. Yes, there's an urgency around what we're doing. The benefits of these, of course, are that we can generate data within a few weeks. But there are, of course, ethical questions around that. The hope around the scenario would be that we would be available to offer immediate medical attention to anyone who might have an effect that would come from post-exposure. But considering that we don't have really a solution, and the potential is the threat of death that could come from a challenge, I don't see that as an ethical approach personally, and I don't think that's the way we really can go about manufacturing vaccines now or into the future. There are other ways to expedite these solutions and other way to test them safely, and I think that's after I guess centuries now, we are able to apply those in an expeditious way to do the right things safely and efficaciously, and within a time frame that properly balances safety with the urgency.
Well, gentlemen, this has been a fascinating conversation. I really appreciate it. If people want to follow your progress, do you have like a website or a Twitter or something like that where you're going to be telling people when you get approved, when you go into animals, etc.?
We have things all over the place at the moment. First of all, the WHO keeps updates on, who are the vaccine candidates at present. We have our own on Theraphage and Metaphage, our own personal sites through the University of Waterloo. So, there are a lot of collaborative components. Everything's tied to anything. So if you go on one site, I'm sure you'll be linked to just about everything you can hope to find.
Well, I wish you all the best. And if you want to come back in a couple of weeks or a couple of months and tell me how you're doing in your animal testing, and whether Emmanuel was actually able to get it up people's noses or not. Let me know, because I'd love to have you guys back and hear more about it. So thank you very much. I really appreciate you joining us tonight.
Thanks so much. Thank you for the invitation. It's been great to be here.
Thank you. Well, this has been a fascinating conversation with three professors and assistant professors at University of Waterloo, talking about building a vaccine. And it's been fascinating. So I really appreciate them joining us. Good night, everybody. Thanks for joining us. Good night.
Good night. Thanks so much.